Today we have the distinct pleasure of welcoming Dr. Khosravi to the sugar science. He is going to be speaking about the role of the thymus in spontaneous development of a multi-organ autoimmune disease in human immune system mice. He's currently an assistant professor at the Department of Immunology at the Mayo Clinic in Arizona. And he comes from doing a postdoc under the supervision of Dr. Megan Sykes at Columbia University. So that's quite a trek from New York in February to Arizona. So it must be a nice something new for you. During his postdoc years, Dr. Khosravi has worked on different approaches of inducing immune tolerance in transplantation and autoimmunity, including thymus transplantation and engineering of Tregs and hematopoietic stem cells using humanized mouse models. He's worked on identifying the mechanism underlying the development of autoimmunity, something near and dear to our hearts here with type 1 diabetes. This is also including the role of thymus and lymphopenia-induced proliferation of T cells. He's also used these models to study the development of human TCR repertoire in human thymus. And currently, Dr. Khosravi's lab research focuses on one, developing chimeric antigen receptor or CAR Tregs with enhanced stability and functionality for inducing immune tolerance in transplantation and xenotransplantation, and two, studying Tregs in autoimmune diseases and applying engineering strategies to develop autologous Treg therapies for these patients. Welcome, Mohsen. Thank you very much for joining us, and I'm very interested to hear about your work and the laboratory you're setting up in Arizona.
Thank you very much. Thank you very much for the introduction, and thanks for the audience to join. I see some familiar faces in the audience. It's good to see you guys. So my name is Mohsen. I'm going to talk about the role of thymus in the spontaneous development of a multi-organ autoimmune disease in human immune system mice. First, let me just introduce the model. Human immune system mice, or more traditionally humanized mice, provides a platform for studying a variety of different human immune diseases. We can study, for example, basic immunology, the development of myeloid and lymphoid cells, hematopoietic stem cell niche, thymic selection processes. We can use these models to study autoimmune diseases like type 1 diabetes, RA, lupus, infectious diseases, transplantation and xenotransplantation, and also cancer. To make humanized mice or human immune system mice, we can have different sources of, for example, thymus. We can have different sources of hematopoietic stem cells and antigen-presenting cells, different sources of T cells and different sources of target cells, like, for example, beta cells differentiated from embryonic stem cells, iPS cells. So it gives us a variety of different choices to study the immune system of interest that we want. So the model that we have been using in our lab, in Dr. Megan Sykes' lab, one of the main models are the human immune system mice that are generated with human fetal hematopoietic stem cells, with or without human fetal thymus. So these mice spontaneously develop a syndrome resembling autoimmunity at varying times post-transplantation, depending on the model that we use. So this disease is characterized by weight loss, hair loss, enlargement of lymphoid organs, and infiltration of T cells and other immune cells in non-lymphoid tissues. This condition, as you can see, a mouse that has this disease here, this condition limits the experimental window for these mice, and we don't know the main cause of the disease. So we try to investigate the mechanisms that drive this autoimmune disease in humanized mice. And as you can see here, I'm showing the percentage of disease-free mice in humanized mice that have a human thymus versus the ones that have a mouse thymus. Okay, so first, I'm going to show you how this disease looks like. So this is the histology of non-lymphoid tissues in affected and non-affected mice. On the upper row, I'm showing the histology in non-affected mice. So you're seeing, for example, salivary glands, skin, liver, lung, and pancreas. And in the lower row, you see the same tissues in affected mice. As I mentioned, they are infiltrated with immune cells. For example, in the skin, we have thickening of the epidermis and infiltration of immune cells into the dermis. Also in the liver, we see these patches of immune cell infiltration. In the lung, the alveolar spaces are filled with immune cells, and even in the pancreas. Although in the pancreas, it's interesting that the islets are kind of spared, but the immune cells are infiltrating the exocrine parts of the pancreas. So these mice do not really develop type 1 diabetes, but they do have other infiltration in other tissues.
That's scary.
So yeah, it's interesting.
And feel just at this moment,
Yeah.
I'm just going to say, if anyone has any questions as we go along, please insert your questions into the chat, or raise your hand.
Yep. We have done some immunofluorescence staining to see what cells are really infiltrating these organs in these mice. So in the liver, for example, we do see infiltration of T cells and macrophages. In the spleen, when these mice have the disease, we do see very enlarged multinucleated macrophage cells in the spleen. And also in the skin, we have the infiltration of CD4, CD8s, macrophages, and even some B cells. And looking at the cytokines in the serum of these mice, we have a little bit higher levels of TNF, but it's not significantly more, but interferon gamma is significantly increased in the serum of mice that are affected and have the disease. Looking at the phenotypes of T cells in affected and non-affected mice. So first of all, we have higher levels of T cells in the spleen, bone marrow, lymph nodes, and even in the skin of these mice that are affected as compared to non-affected mice. They have much higher levels of T cells. And these T cells, both CD4s and CD8 T cells, the majority of them, when the mice are affected, are not naive anymore. So we lose a population of naive CD4 and CD8s in different tissues. And the majority of these T cells are now becoming effector memory cells, again, in across different tissues of these mice. So one question was whether this is a, so it looks like T cells are driving the disease here. However, we didn't know whether it's clonal disease or a few clones of T cells are affecting different tissues. So we did single cell sequencing of T cells derived from different tissues of these mice. And we do see that certain clones of T cells are highly expanded. So I'm for example, showing pancreatic islets, skin, liver, spleen, and native mouse thymus of these mice. And these colored areas, each one of them show one clone of T cells based on the TCR alpha and beta, and that is highly expanded and are actually shared between different tissues. So certain clones of T cells in these mice are highly expanded and are affecting different tissues that are targets of the disease. However, in the grafted human thymus, we do not see these highly expanded T cells. So the grafted thymus single positive CD4 and single positive CD8 contains a lot of T cells that are not expanded. So we wanted to study the role of thymus in development of the disease. So before studying that, we had to develop a rapid method for removal of native mouse thymus, because we knew that native mouse thymus, NSG mice we use usually for these models, do support human thymopoiesis. So in order to, for example, when we want to transplant a human thymus to have human T cells only develop in human thymus, we have to remove the native mouse thymus. So we did develop this model, which provides us a good tool to study human thymopoiesis only in human thymus or any other thymus. And using this model, this surgical method, we developed 3 different models of humanized mice. So humanized mice that have no thymus. So in order to do that, we first remove the native mouse thymus, irradiate the mice, and then inject them with human fetal liver CD34 positive hematopoietic stem cells, and then follow these mice with checking human immune cell reconstitution, and finally euthanizing the mice at the end of the study. To study the mice that only have human thymus on board, we again remove the native mouse thymus, do the same procedures. However, we also transplant these mice with autologous human fetal thymus. So we cut these human fetal thymi into small pieces of 1 to 2 millimeter cubic and transplant them under the kidney capsule. And we also take some measures in order to remove the pre-existing thymocytes from these mice. So the majority of T cells that develop in these thymi are newly developed T cells that come from the hematopoietic stem cells. And a third model here is when we study the disease in mice that have mouse thymus on board. So we do not do any thymectomy in this model. We just leave the native mouse thymus there, but do not transplant any human thymus. So when we compare and follow these three models of humanized mice, we do see that mice with no thymus do not develop the disease. So T cells are absolutely required for this autoimmune disease to develop. Mice with the mouse thymus have a faster disease development as compared to the mice with the human thymus. So we wanted to see why is that, and why having a human thymus on board kind of delays the disease as compared to the mice that have only murine thymus. So first we looked at the structure and the histology of native mouse thymus and grafted human thymus. So when we transplant human thymus under the kidney capsule, it becomes like this, almost the size of the kidney. So it grows a lot as compared to the native mouse thymus, which is very small and vestigial. So if we compare the NOD thymus, the right lobe and the left lobe of the thymus, it's much larger than the NSG mouse thymus. So NSG is on the background of NOD. So NSG mice is NOD mice that are skewed and have common gamma chain knockout. So they have much.
Excuse me, was that a surprise that it grew so large?
So this is the normal size of the NOD mouse thymus. So it was a surprise.
No, the grafted. You said it grew much larger.
Oh, do you mean the grafted human thymus?
Yes. Mm-hmm.
No, that's what we usually see. So
okay. I just wanted to clarify that.
Yeah, no, that's the usual. I mean, but when we transplant these thymi under the kidney capsule, they're very small, actually much smaller than the native mouse thymus, but they grow a lot. And it's only specific to fetal thymus. So if you transplant postnatal thymus, it does not grow that much. So that's actually another interesting story that our lab is pursuing. But we use human fetal thymus because we know it grows and it supports. And when you look at the structure of these grafted human fetal thymus, you do see these different areas of thymus. I mean, the cortex, the medulla, basal corpuscles. So it actually forms a very well-structured human thymus.
That's fascinating.
And yeah, and even the immunofluorescence staining shows, for example, we checked HLA-DR, cytokeratin 8 and cytokeratin 14. Cytokeratin 8 mainly characterizes the areas that are cortical areas of the thymus, and while cytokeratin 14 mainly shows the medullary areas plus the HLA-DR. So the same when you do
one last question. We have one question in the chat. What is the difference in thymus size in comparison to B6 mice? NSG doesn't have an immune system. Can you field that one?
Sorry, as compared to black 6 mice, you said?
Yeah. What is the difference in thymus size in comparison to B6 mice? NSG doesn't have an immune system.
Right. So the NSG mouse thymus is very vestigial and very small. I don't know the comparison between NOD and B6. I believe both of them are big and similar size. But since the NSG, I mean, there is no development of T cells, murine T cells in this NSG mouse thymus. But when we transplant it with human thymus, with human hematopoietic stem cells, there will be human thymocytes developing in this mouse thymus. But still, it does not develop a very well structured. So this is, for example, one NSG mouse thymus, the native mouse thymus that has human thymocytes, but still it does not form the cortical and medullary areas that we see, for example, in grafted human thymus. So, and then when we stain, for example, for cytokeratin 8, and UEA1 is a marker of MTEX, so we do not see actually any MTEX in these native mouse thymi in NSG mice, as compared, for example, to NOD mouse thymus that shows the areas of, for example, these areas are medulla that contain UEA1 positive cells, but in NSG mouse we don't see them. So looks like we don't have really a well organized structure here, but the percentage of double positive and single positive CD4 and CD8s are very similar between grafted human thymus and native mouse thymus. However, the level of Tregs are lower in the native mouse thymus as compared to grafted human thymus. So looks like there is some lack of structure here, and we hypothesize that this lack of structure might lead to leakiness of these native mouse thymi to autoreactive T cells, and probably that's why that these mice develop a faster disease in humanized mice. So to test that, we used a system that was previously developed in Dr. Crusoe's lab, which in our center. So he has previously developed this model with MART1 TCR transduced hematopoietic stem cells, and also transducing hematopoietic stem cells with MART1 peptide. So before transplanting the hematopoietic stem cells to these mice, we transduce them using lentiviral vectors. We transduce them, half of them with MART1 TCR. So this is an HLA-A2 restricted TCR that recognizes the melan A antigen or MART1. And the other half of hematopoietic stem cells, we either transduce them with the MART1 peptide or did not transduce them. So we wanted to see what happens to the selection of this TCR if the cognate peptide is presented in the thymus. So we were expecting that the presence of the peptide leads to negative selection of this TCR. So we tested this system, this model, in native mouse thymus versus grafted human thymus, and then looked at double positive thymocytes, single positive CD8s, and single positive CD4s, and stained them with the tetramer that detects the MART1 TCR. So in the absence of MART1 peptide, this TCR is positively selected in the grafted human thymus. So it is positive in double positive and single positive stages. However, when the peptide is presented in the thymus, by transducing the hematopoietic stem cells, the dendritic cells will take up the peptide and present it to the thymocytes in the thymus. We do see that this TCR is negatively selected. So we have an absolute deletion of MART1 TCR T cells in the grafted human thymus. However, when we look at the native mouse thymus, it's not the case. So in the presence and absence of the MART1 peptide, we still do see MART1 TCR transduced MART1 TCR T cells present in the native mouse thymus, which shows to us that this lack of structure in the native mouse thymus leads to lack of negative selection of this TCR, even in the presence of the peptide. So that that supports our hypothesis that the lack of structure in the native mouse thymus leads to leakiness of native mouse thymus and potentially could lead to the autoimmune development.
Can I just ask, do you
right.
see in humans that progress to or that are at risk for autoimmune disease like type 1, with they have the HLA risk factors, is there any kind of disorganization in their thymus in any of the tissue samples that are seen? If there's, this is prior to what, like 10 years old, because the thymus disappears.
Right. So I believe we still don't know the answer as far as I know. And not a lot of studies have been done. And I believe that's the field of study that requires more investigation. And it's a little bit difficult because you cannot really sample the thymus. The only way to analyze the thymus probably is with sonography to test maybe the size of the thymus. And also, the other thing that we can do is to measure the level of recent thymic immigrants in the periphery of those patients. There are some suggestions in the literature that in certain autoimmune diseases, including type 1 diabetes, there is lower levels of recent thymic immigrants in the peripheral blood of these patients, suggesting that there might be some differences in the size or structure of the thymus. But probably humanized mice actually provide a good platform for studying this question. Actually, our lab is currently working on. I mean, like Megan Sykes' lab is currently has, and also Dr. Crusoe's lab. They are working on this to study the role of thymus in type 1 diabetes specifically, with a new project under the head.
No, that's fantastic. It's a great model system. And
Yeah.
as we know, Stan Wang in Boston just launched his company Thymune with some ideas in that same kind of vein. So it really it's kind of interesting that the thymus is coming back, not coming back, but getting more attention, more love these days as a potential target.
Exactly. So so we hypothesized that, I mean, we were wondering why is that the native mouse thymus in NSG mice have this problem, a lack of structure. So one hypothesis that we had was that maybe a lack of interaction, early in life interaction of hematopoietic cells and the thymic epithelial cells might be the reason that these thymi do not form a very well structure. Because we know that thymic epithelial cells require interaction with hematopoietic cells and thymocytes for fully developing functional thymic epithelial cells. So, so we decided to study whether injection of hematopoietic stem cells at the neonatal stage versus an adult stage might make any difference in development of the disease and formation of a native mouse thymus structure. So we injected hematopoietic stem cells in the neonatal stage 2 or 3 days after birth, versus in the adult stage 6 or 7 weeks after birth of these mice, and compared the disease development. And we found that injection of neonatal injection of HSCs to some extent or significantly delayed the disease development in these mice. However, the number of thymocytes was not different between these two groups. And when we looked at the structure of native mouse thymi in these two groups, it was not really different. And still, injection of hematopoietic stem cell in the neonatal stage did not make the structure any better. So probably it's not the early interaction. So we have our own hypothesis regarding that I will say at the end. The other thing that we did to test the mechanism in this model was to test whether presentation of mouse antigens by murine MHCs is contributing to disease development, or whether it's an indirect presentation of mouse antigens by human APCs that can drive the disease. To address this, we studied the disease development in pure NSG mice versus NSG mice that are knocked out for murine MHC class 1 and class 2. So in this model, there is no murine MHC present there to present mouse antigens. So if the disease was delayed in this model, it could show to us that the presence of the presentation of antigens by murine MHCs is driving the disease, but that was not the case. So the disease development was very similar in NSG mice versus NSG mice that are knocked out for murine MHC class 1 and class 2, which is showing to us that the major driver of the disease is the indirect presentation of antigens by human antigen-presenting cells, not murine MHCs. So in conclusion, lymphoid and non-lymphoid organs of affected mice contain mainly memory type human T cells. Both CD4 and CD8 T cells become activated and infiltrate the target organs. Thymus is required for autoimmunity development in this model. Mice with human thymus showed delayed onset of the disease and longer survival compared to the ones with the murine thymus. The native thymus in NSG mice do not develop a normal structure, which leads to impaired thymic selection. Neonatal injection of human hematopoietic stem cells delays the disease, but does not allow normal mouse thymus structure to develop. An indirect presentation of mouse antigens but by human dendritic cells can drive the disease. So I wanted to say at the end, what we think is probably the real reason that these mice do not, the NSG mice do not form a very well-structured murine thymus is probably due to the lack of common gamma chain and IL-7 signaling, because we know that common gamma chain is required as part of the structure for IL-7 receptor as well. So these mice do not have IL-7 signaling, and we know that IL-7 signaling is required for tissue inducer cells that are the precursor cells for formation of lymphoid organs, lymphoid tissues, including the thymus. So that is our current hypothesis, that probably lack of IL-7 signaling is the major driver of this condition in these mice. So I think I'm gonna end the presentation here. I had another story about lymphopenia-induced proliferation, but I don't think we have enough time and to discuss it at this point. Hopefully in the future we can discuss that story as well. At the end, I would like to thank my previous mentor, Dr. Megan Sykes. It was great to work in her lab for a couple of years, and my colleagues in the lab that are listed here, and other labs at our center in CCTI, lab of Dr. Winchester and Dr. Remy Crusoe, and our flow cytometry core, and also the funding agencies that help us do this type of research. And as you mentioned at the beginning, I have recently moved to Mayo Clinic. Actually, I started my work here last week, and I'm currently looking for 2 postdocs. So we are in the Department of Immunology at Mayo Clinic, Arizona. So we want to have 2 enthusiastic postdocs to work in the field of transplantation and autoimmunity. As you mentioned, we are working on chimeric antigen receptor or CAR Tregs in transplantation and xenotransplantation. We are using these humanized mouse models that I explained to study the rejection in humanized mice and testing these CAR Tregs for prevention of rejection. And in the second study, we are characterizing regulatory T cells or Tregs in patients with autoimmunity, and we want to see if we can later do some genetic manipulation and corrections to develop a autologous Treg therapy for patients with autoimmunity. Thank you very much.
Those projects looks really exciting. And we are posting them on our jobs board at the Sugar Science. So hopefully people will take a look at that and apply. I wanted just to clarify, are you in Phoenix or Scottsdale? I know the Mayo Clinic is in both locations.
So the lab is in Scottsdale. So Mayo Clinic here has actually 2 sites. So the research campus is mainly in Scottsdale, but there are research labs also in the Phoenix campus. The Phoenix campus has the big hospital there. There's a lot of collaboration between the 2 sites, but we are located in the Scottsdale campus.
Scottsdale is beautiful, so that's beautiful. Fantastic. I have one comment here from the audience. Great talk. Have you tried mouse fetal thymus transplantation? Is the question.
Yes, actually, I tried B6 and NOD mouse thymus transplantation. Unfortunately, due to the technical issues, we did not succeed at the time because, again, you need to have I believe around day 14 was that in the literature was shown that thymus, you need to take it at, I mean, the embryonic stage, day 14. That's the time that you need to take the thymus, murine thymus, to transplant. And there are some videos in the literature that we tried to replicate that and get the thymus. It was very small, and there are a lot of tissues that are very similar to the thymus. So we harvested some thymus, transplanted, but unfortunately they did not grow and support thymopoiesis. So that was the issue. But we needed more expertise in doing those types of transplantation, murine thymus transplantation.
Okay, well, perhaps that's on the menu maybe for the future in the Khosravi lab. It was very nice speaking with you. Fascinating work, and really two exciting projects for some postdocs to come and dig into. So hopefully you'll be getting some applications soon. And so I hope you have a great rest of the day, and thanks again, and wishing you all the best.
Thank you very much, and I appreciate you and all the audience that joined. Thanks a lot. Have a good day.
Great. You too.